Welcome to AP Biology. My name is Paula Dunker. In this lesson, I'm going to discuss with you the directionality of the subunits of proteins, the four elements of protein structure, and how a change in the subunits of a protein may lead to changes in its structure or function. Proteins are comprised of linear chains of amino acids, and in Figure 1, we see a single amino acid. If you look at the central carbon, coming off to the left, we have an amino group, NH2, and to the right, we have a carboxyl group, COOH. So we have two distinct ends to this amino acid, giving the amino acid directionality with an amino terminus and a carboxyl terminus. And as proteins are synthesized, new amino acids are added to the carboxyl group of the existing amino acid chain, forming peptide bonds or covalent bonds between the carboxyl group you see here of the left amino acid and the amino group of the amino acid on the right. As you continue to add more and more amino acids together, we get a linear chain of amino acids as shown here in Figure 2 with two distinct ends, the carboxyl and the amino terminus. And if new amino acids are going to be added to this long chain, they will be added to the carboxyl terminus. This linear chain of amino acids determines the primary level of structure for a protein. And there are four elements of protein structure, starting with this primary level, which is determined by the sequence of amino acids held together by covalent bonds. The proteins are not yet functional at this level, and additional folding is needed. So next we have a secondary level of structure that arises through local folding of amino acid chains into elements such as your alpha helix and your beta sheet. And I'll model this for you in the next slide. But the protein still needs to have additional folding occur before it's functional. So we have the tertiary structure, which is the overall 3D shape of the protein and often minimizes free energy. So this is the more stable level of structure for proteins, and many proteins will function at this tertiary level. And the tertiary level comes about through various types of bonds and interactions, particularly with the R groups of amino acids that could vary in chemical properties based on the side chains that are linked there. Some proteins, however, do need to reach a fourth level of structure called the quaternary structure, which arises from interactions between multiple polypeptide units. So in this image, we have three whole polypeptide chains working together. Hemoglobin is a good example of a protein that reaches this quaternary structure. It is a protein found in red blood cells that transports oxygen. But I want to just stress here before we move on that regardless of whatever the 3D shape and functionality of that protein is, it all starts with that primary level and that sequence of amino acids and the chemical properties that each of those possess. So here in this video, I'm going to model for you the folding so you can see it all in action using a pipe cleaner and some beads. The pipe cleaner represents the carbon backbone of the primary level where you have the peptide bonds holding amino acids together between that carboxyl terminus and the amino terminus. And the different color beads represent the properties of the amino acids. So red beads are nonpolar because they have nonpolar R groups, and blue beads are polar because they have polar R groups. So next we're going to see that local folding happen. So here you see me folding it back and forth, giving rise to the beta sheet element shown there. And then we're going to have some alpha helix action happening on the right. Okay, so now we have our secondary level of structure with those two elements, the beta sheet to the left and the alpha helix to the right. But the protein's still not yet functional. We need to have some additional folding into that 3D tertiary shape. So let's assume that we have some water here in the surrounding environment that this folding's occurring in. The red beads are nonpolar, so they are hydrophobic, and they're going to want to fold inward towards the core of this protein. So in the core, we're going to get these hydrophobic interactions occurring since there's water in that environment. The blue beads, which are polar amino acids, they're going to be hydrophilic and interact with water. So they're going to fold towards the outside of this protein as we start to take this 3D shape. And so you see that here in our model. So here, this would be the tertiary 3D form, which could be functional for many proteins. If we want to model quaternary structure, then we need to have the interaction of two or more whole polypeptide chains. So here is an additional polypeptide chain interacting with our original one. So this would be quaternary structure. So let's focus though on our tertiary protein here. Let's say this is a hypothetical folding of a protein into a receptor protein. Receptor proteins are responsible for receiving chemical signals. The Play-Doh ball here can represent our chemical signal. The signal molecule must have a similar structure as the receptor in order to connect to the receptor. So we're going to use this hypothetical protein to practice our skill of argumentation. So argumentations are important for scientists to make, and making predictions are an important component of an argument. So predictions are expected outcomes based on changes or disruptions to one or more components in a biological system. So let's see how this might impact our hypothetical protein. So in this image you see here, let's say that the instructions for assembling this protein were changed, and this polar amino acid will be switched out for a non-polar amino acid. If we look at our question, predict the change in the compatibility between the receptor protein and the signal molecule as a result of the change in amino acid, and predict the change in the function of the receptor protein. So you're being asked to make a prediction on 2 items, so be sure you address both of those in your prediction. So I'm going to ask you now to pause the video, take a moment to think about what might be a good prediction for this question, and then press play and we'll discuss the answer together. Okay, so hopefully you have your prediction ready, and let's see what happens if we have that change. So here we're changing that polar amino acid out for a non-polar amino acid in that primary level of structure, which leads to this tertiary folding eventually. So now we have a hydrophobic non-polar amino acid that will not want to interact with the water, so it would have folded into our core, along with those other hydrophobic interactions. So in terms of the first part of our prediction, compatibility, the shape of the receptor protein will change, and therefore the protein will not be compatible with the signal molecule, as you see here. So that's the first part of our prediction. As for the second part of the prediction, the function of the protein will also be changed, and it will no longer be able to bind to the signal molecule and receive the signal as shown in the video. So we have those 2 components addressed. The compatibility will change and it will no longer be compatible, and the function will also be changed and it will not be able to receive that signal effectively. So here are some key takeaways. Amino acids have directionality with an amino terminus and a carboxyl terminus. Amino acids are added to the carboxyl terminus of a growing peptide chain, forming covalent bonds. There are 4 elements to protein structure: the primary, which is that linear sequence of amino acids; secondary, your alpha helices and beta sheets; tertiary, your 3D functional form for many proteins; and quaternary, where you have multiple polypeptide chains interacting. The resulting 3D shape gives rise to the protein-specific function, and a change in an amino acid subunit at that primary level of structure may lead to a change in the structure and function of the protein. If you want to explore more protein folding, I've shared with you this really cool protein folding interactive found at lab.concord.org at the website address given. Thank you for watching, and be sure to check out video lesson 3, where I'll discuss the structure and function of carbohydrates.
Welcome to AP Biology, topic 1.7, proteins. My name is Linda Aloju, coming from you from Plano, Texas. In this daily video number 2, we are going to discuss the directionality of the subunits of proteins, the 4 elements of protein structure, and how a change in the subunits of a protein may lead to changes in its structure or function. So what are we going to learn in this video? We're going to learn 5 things. The first thing is, what type of bonds connect amino acids to ultimately form proteins? Number 2, what are the components of an amino acid, and how do R groups influence protein structure and function? Number 3, how does the sequence of amino acids determine a protein's shape? Number 4, how do hydrogens contribute to the secondary structure of proteins? And finally, number 5, how do different interactions among polypeptides contribute to a protein's quaternary structure, and why are these structures important for a protein's function? So let's take a look at the monomer, that building block of proteins. That is going to be an amino acid. So amino acids are composed of a central carbon with a hydrogen atom. It also has a carboxyl group, it's going to have an amino group, and it's going to have a variable R group covalently bond to it. So we're going to go into these R group interactions later, but our R groups, they have properties that are similar. So they're going to have hydrophobic or non-polar properties. They can have ionic properties. They can have disulfide bridges, and they also can be hydrophilic or polar in their interaction. Okay, now that we know the structure of amino acids, we can now put them together, link them together to make that growing polypeptide chain. So those proteins are composed of linear chains of amino acids connected by the formation of a covalent. That covalent bond is going to be called a peptide bond between the carboxyl group of one amino acid and the amine group of another amino acid. So there's going to be that peptide bond. Again, you can see in the image below in the left one, there's going to be that carboxyl group. In the right green, we have that amino group. Together, they're going to lose that water through dehydration synthesis. So as that water is removed, we can join the two amino acids together and make that peptide covalent bond to form that growing peptide chain. So now that we have that growing peptide chain, that specific sequence of amino acids and proteins will determine its overall shape. So the shape of a protein, they're going to have to first go through that primary structure, that secondary structure, that tertiary structure, and sometimes that quaternary structure. So let's take a closer look at these different levels of protein folding. So the primary structure of proteins is that simple chain of amino acids that are linked together by those covalent or peptide bonds. So just as a reminder, again, the primary structure, we're going to have one amino acid. The carboxyl of one amino acid is going to covalently bind to the amine group of another amino acid to form that covalent peptide bond that will then link them together to form this linear growing polypeptide chain. So now we're going to start getting a little bit of that dimension to the folding. So we're going to get to that secondary structure. So in our secondary structure, as you can see in the image, right, we have that peptide bond between the carboxyl and that amine group. So now what's left over is there is an oxygen in one amino acid and a hydrogen in another amino acid. So now those two are going to hydrogen bond between the atoms of the polypeptide backbone of the amino acid side chain. So those are going to hydrogen bond with each other, that oxygen and that hydrogen. Now, how it's going to bind with each other will then determine its shape. So if it's twisting around that backbone, we're going to form that alpha helix as our secondary structure. But if we are kind of folding back and forth with those hydrogen bonds, that's going to form that secondary structure of a beta pleated sheet. So now that we have the primary and the secondary structure, we can get to our tertiary structure. This is when my protein has that three-dimensional shape of the tertiary structure of a protein resulting from the formation of my different R group interactions, those hydrogen bonds, those hydrophobic interactions, those ionic interactions, or those disulfide bridges. So again, that R group interactions is going to drive the folding of this tertiary structure to give it that 3D shape. Sometimes we'll have quaternary structures. So the quaternary structure arises from interactions between multiple polypeptides. So this is more than one polypeptide chain folded in its tertiary structure to now come together to form that quaternary shape. All four levels of protein structure determine the function of a protein because, again, those amino acid R group interactions are going to determine how it folds. So let's practice. This free response question is centered around science practice number 6, predict something when something has changed in the scenario. So let's take a look at this question. It says hemoglobin is a protein with a quaternary structure composed of 4 polypeptide subunits. Each subunit contains an iron-containing heme group that binds to oxygen. The quaternary structure of a hemoglobin is crucial for its ability to pick up oxygen in the lungs and release it in tissues where oxygen is needed. Predict what would happen to a hemoglobin's ability to bind and release oxygen if a mutation caused the loss of one of its subunits. Take a moment and think about your answer. Okay, so what is this question asking us? It's asking us to predict what would happen if a mutation was there, right? So in this question 6E, there's a disruption, there's a change. So now a mutation has been introduced, which will now affect the subunits. We're going to lose a subunit. So what's going to happen if we lose a subunit in that quaternary shape? So one of those subunits won't be there. It's going to affect the way it folds. So we can say that the hemoglobin's ability to bind and release oxygen is going to be less. with this mutation. So what should we take away from this video? Some key takeaways. The first thing is proteins are made of linear chains of amino acids that are connected by covalent bonds, and that particular name for that, again, is that peptide bond. We're also going to take away that each amino acid has a central carbon, a hydrogen, our carboxyl group, an amine group, and a variable R group that affects its properties. We're going to also take away that the sequence of amino acids determines a protein's primary structure and then its overall shape. We're going to take away that secondary structures such as alpha helices and beta pleated sheets are formed through that hydrogen bonding around that polypeptide backbone. And that the remaining R group interactions, those remaining R group interactions is how the tertiary structure results from various interactions. Again, those hydrogen bonds, those hydrophobic interactions where they're going to want to be on the inside, that ionic interactions and disulfide bridges. Lastly, we're going to take away that quaternary structure forms when multiple polypeptides interact. So more than one polypeptide come together, and all 4 levels of structure determines a protein's function. Thank you so much.